reproductive system, we know that there are a number of different organs that we're going to be looking at histologically. We have the two testes, and we have a tubular system that we're going to look at in some detail, so we'll see that. The two testes are in a scrotal sac, and then the tubular system that we'll look at is essentially we go from some structures called the seminiferous tubules to the rete testes to the ductus efferentis to the epididymis to the vas deferens, which is also called the ductus deferens. And then we have some accessory structures that are going to add to the semen, the two seminal vesicles and the prostate gland and the bulbourethral gland. And so we'll also look at the urethra, the prosthetic urethra, and then the urethra coming down through the penis. And so we're going to be looking at the histology of all these different structures. We know that genetic sex is determined if there is an XY chromosome, then the presence of an SRY gene on the Y chromosome causes these indifferent gonads to differentiate into the testes with supporting Sertoli cells and Leydig cells, which produce the androgens, the testosterone. And the Sertoli cells, one of their many functions is to cause regression of the Mullerian ducts with Mullerian inhibitory substance, and the testosterone helps the indifferent gonads transform into the male reproductive tract. And if it's an XX chromosome, there is no SRY gene. In the absence of the androgens, testes determining factor, and then the antigens, the gonads go into a female pattern. There's an absence of Mullerian inhibitory substance, and so the Mullerian ducts transport into the female reproductive tract. The absence of testosterone causes the Wolffian ducts to regress, and so it turns female. So you remember all that, but that's a quick reminder for us. When we look at the testes are the male gonads. You remember that the gonads, testes in male and ovaries in females, have two primary purposes. One is meiosis, either spermatogenesis or oogenesis. And the other is production of the reproductive hormones. In males, that's primarily the androgens, which is mostly testosterone. In females, that's estrogen and progesterone especially. So the testes are going to do spermatogenesis, production of the sperm, in other words. Spermiogenesis, which is that last, after spermatogenesis has happened, the maturation of the sperm cell is called spermiogenesis. So that all happens in the testes. Again, the testes, if we look at the testes, we see that we have these different lobules. So like here's a lobule, and here's a lobule, and here's a lobule, and here's a lobule. There's some septa or trabeculae that come from the tunica albuginea out here that separates the testes into these lobules. Within the lobules, we have the seminiferous tubules. That's these coiled tubules that we see here. And also, we have some interstitial cells of Leydig, which are the cells that produce the testosterone. This histology slide down here is from your Loma Linda slide set. These are the seminiferous tubules. And in the lumen of the seminiferous tubules is where spermatogenesis occurs. That process is aided by the supporting cells, which are called the Sertoli cells. Outside of the seminiferous tubules, we have the interstitial cells of Leydig, which produce the androgens, which is primarily testosterone. When the seminiferous tubules produce the sperm, next it goes to this area right here within the testes. That's the rete testes. This area, the rete testes, is in an area called the mediastinum. So you have the mediastinum here, but the tubular structure in here is the rete testes. This then goes to the efferent ducts, the straight ductules, which goes into the epididymis. The sperm is stored for often a long time in the epididymis, and that's where it's fully gets full maturation and capacitation. And then it goes on through the vas deferens and the efferent ductules and on into the body. And we'll look at the rest of the reproductive tract a bit later. This is a little diagrammatic representation of this spermatogenic process, the formation of the sperm and the seminiferous tubules. Here again is a little bit more, this is from your book, this diagram. This shows you the lobules with the seminiferous tubules, the rete testes, the straight ductules or the efferent ductules, and then the epididymis, and then the vas deferens or ductus deferens. This would be the scrotal sac coming around, and we have the so-called tunica vaginalis. This double membrane coat is named for the female reproductive system. So it's called tunica vaginalis in both males and females. It has a double layered membrane. The outer layer is the parietal layer of the tunica vaginalis, and you can see that layer around here, coming around, and then the inner layer, as it continues in and attaches to the actual wall of the testes, that's called the tunica vaginalis visceral layer. And then we have the potential or serous space between the two layers. So double layered membrane there. Now just deep to the visceral layer of the tunica vaginalis, you can see that purple line there, just deep to that is the tunica albuginea. And so there it is, the tunica albuginea. And invaginations of that tunica albuginea are the septa, or sometimes they're called trabeculae, but septa, which divide the testes into these lobules. And then we have the seminiferous tubules within the lobules. This shows it again, the two different layers of the tunica vaginalis that we see here, the parietal layer being more superficial, and the visceral layer of the tunica vaginalis being right around the outer part of the testes. And then we have the tunica albuginea, and here's the lobules. This is an actual from a rat, and it shows you some. No, this isn't from a rat. That would be an impressive rat. This shows it with a human with the different structures in a type of like a fluorescent kind of imaging. The seminiferous tubule has been pulled out here just to show that it's coiled, highly coiled here, and then the rete testes and the straight ducts, the ductus deferens, and the epididymis, the vas deferens coming around, and we'll see these other structures. Here it is again. We're getting used to this kind of a diagram, so we won't belabor it too much. Here it is in the actual histological image. This is very low magnification. magnification. We do see the tunica albuginea. At a higher magnification, we might well see the visceral layer of the tunica vaginalis as well. Generally, in our histological slides, the outer layer of the tunica vaginalis and the whole scrotum is missing, so we're not really going to see that. But we'll see the rest of it. Here we have the lobules and the seminiferous tubules. This area in the middle here, kind of, that I'm outlining with the laser pointer, this is the mediastinum, and housed within that we have the rete testes, that's what the RT is indicating. And then those will go to the straight ducts, which will go to the epididymis. Notice the epididymis is really entirely outside of the testes. So we'll be looking at this, of course, at much higher magnification. This is still fairly low magnification, but now we're seeing the seminiferous tubules within the testes. So each of these is part of that. A lot of these could be the same tubule, but they're very highly coiled, and so when you cut across them, you might get the same tubule more than once. The lumen of the seminiferous tubules is the epithelium where spermatogenesis occurs. The less mature cells are to the outer parts of the lumen of the seminiferous tubules. As you go deeper towards the center, you get more and more mature cells. So the sperm cells would be these here. And we're going to have multiple slides looking at these and identifying these different cells types. In addition, these cells outside of the seminiferous tubules, such as here that we're tracing, and here, and here, and here, and here, these are the interstitial cells of Leydig. They tend to have a fairly eosinophilic cytoplasm with a fairly darkly, intensely stained nucleus. In addition, we might have some of these fibroblasts supporting cells that are flatter, and some of these, especially around the periphery right of the seminiferous tubule, can be called myofibroblasts because they have some contractile ability. They're a little bit of a variant of the fibroblast.
So are those Leydig cells, those are the ones that secrete the testosterone?
Exactly.
Okay.
So these are endocrine cells.
And that happened, so that is that technically the secretion of that happening outside of the seminiferous tubules?
It goes into the blood.
Okay.
So this is a hormone, so it's going to go into blood vessels in the testes and then just be carried away from the blood. All these other cells, the sperm, they're going to be carried away through the ducts.
Mm-hmm.
But the androgens, the testosterone, essentially is going to go into the blood and be carried throughout the body in that manner.
And are the seminiferous tubules, like all those little black dots, are those Sertoli cells?
All these little black dots are a variety of cells.
Okay.
They are spermatogonian, they are primary spermatocytes, secondary spermatocytes, spermatids, and sperm, and Sertoli cells.
Okay.
So when we get it in a higher magnification, we're actually going to be able to tell and differentiate all these, which is a lot of what we're going to do right now today. So we're going to get a lot of practice. I probably went overboard with the number of seminiferous tubules that we're going to show you with these different cells. And they're not too hard to identify. This is just a little collagen, connective tissue and all that as well in the supporting. Just a quick reminder, the stem cells, the undifferentiated cells, are called spermatogonia. Spermatogonium is singular, spermatogonia. They can divide and increase in their number by mitosis, in other words, simple cell division. But some of these spermatogonia can differentiate without dividing into primary spermatocytes. Notice these are diploid with the 2N still. The DNA has already doubled before you go through this process. So the primary spermatocytes, when we say 2N, it's diploid, but they're double-stranded chromosomes. You've already doubled your DNA.
But the spermatogonia, they do not have the double.
Spermatogonia double as, yeah, I would say that's true.
Okay.
They're doubling as they're turning in, differentiating into primary spermatocytes.
Okay.
So that is happening, doubling, but there's no cell division as part of that doubling.
Okay.
You're doubling it as a precursor before you go through the rest of the process of spermatogenesis. Now, as the primary spermatocytes, they will then divide the first cell division into 2 secondary spermatocytes. Notice that each daughter cell, each secondary spermatocyte is now 1N. They're haploid. But we need to remember that although there are 23 chromosomes in each daughter cell, they both have double-stranded DNA still, chromosomes. Okay? So it's 23 chromosomes, but there are 23 double-stranded chromosomes in each secondary spermatocyte. Each secondary spermatocyte then divides into 2 additional daughter cells, resulting in 4 compared to your primary spermatocyte. And those double-stranded chromosomes each divide, so now you have 23 single-stranded chromosomes in each of these daughter cells. So they're still haploid, even though you've had another cell division. So both the secondary spermatocytes and the primary, and the spermatids, are haploid. But the secondary spermatocytes are double-stranded chromosomes, haploid, and the spermatids are single-stranded chromosomes, haploid. Now that's technically the end of spermatogenesis. After the spermatids are formed, we have the differentiation of the spermatids into sperm, and that is called spermiogenesis. There's no cell division there. It's just the cells take a different form. The cells, for instance, grow a tail, a flagellum, and the head forms and all of that. So the spermatids differentiating into the spermatozoa is called spermiogenesis. Another thing to note here, so it won't confuse you, all of the cells here, plus the Sertoli cells, we're going to see histologically, except the secondary spermatocytes. As soon as the secondary spermatocytes form, they start dividing. And so we really don't see secondary spermatocytes. We know they're there, but we know they exist, but they don't hang around long enough to get their picture taken, essentially. And so we don't really see these to identify. Let's see, is it worth watching this? Yeah, I guess.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. Secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
Well, that's essentially a quick review of that. Okay, now we're going to look at several images of this. This just need to warn you that this is a 640 magnification. This is from the book, this picture. We're gonna see it at 400, so they're not gonna look this big. And so, just to let you know that. Now, here we are, this is the seminiferous tubules. These are the spermatogonia. Now, you can divide those into A and B, but we're not gonna worry about differentiating them. It's very difficult to differentiate them, especially at the magnifications we're gonna be looking at. So these are the spermatogonia. They tend to be outside, to the outside edge of the seminiferous tubule. We could say it's right under the basement membrane or the basal lamina. These are the spermatogonia. Now, as we go a little deeper, when we see this stringy chromosomal-like structure in cells, such as here, and 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 here, these are the primary spermatocytes. Okay? Those are the primary spermatocytes. We don't see the secondary spermatocytes, so the next one we see are the spermatids, and all of these here are the spermatids, and then we see the sperm cells. Again, the deeper you go towards the lumen, the more mature your cells are. In addition to those cells of spermatogenesis, we have the supporting cells, which are called the Sertoli cells. They tend to be elongated, but they can have a kind of a triangular shaped nucleus, but the cells tend to be elongated very often, and you have a prominent nucleolus, as you can see there. So that's a Sertoli, that's a Sertoli, that's a Sertoli. Here's one here, here's one here, and here's one here. There's a bunch of them, okay? There's one, there's one. These are the support cells, and we'll get into a little bit more what their function is. But part of their function is they form a blood-testes barrier, so that these gamete cells that are being formed, the sperm, are not susceptible to attack by the person's own immune system. You have to protect them from the immune system. And the Sertoli cells do that. The hormone FSH, follicle-stimulating hormone, and testosterone from the interstitial cells of Leydig both bind to these Sertoli cells, and that stimulates and helps the Sertoli cells guide the spermatogenic process. The Sertoli cells also remove defective sperm cells, for instance, and we'll look at some of the other features that they do, but those are some of the things that they do. We see some somewhat modified fibroblasts. These are myofibroblasts. You can see the name here, myofibroblasts. And again, these are a type of fibroblast that has some contractile ability, and that contractile ability in this case helps push the sperm along their way out of the seminiferous tubules. This is sort of that pathway that's happening. These are the Sertoli cells, diagrammatically represented. Here's your basement membrane or basal lamina. And you take your spermatogonia and they differentiate all the way down with the help, with the aid of the Sertoli cells. You're quite aware, I'm sure, that spermatogenesis is temperature dependent. And if the temperature is too high, then the sperm, not enough sperm may be produced, oligospermia. And that, of course, is why the testes are outside of the body proper for that reason. This is a pretty nice looking plastinated view of it. And we can see the primary spermatocytes really well here. And you see all of these stringy type of chromosomes. And then we have a number, here's some that are very easy to see. These are the spermatogonia. See out here? And there's some over here on this other seminiferous tubule. So these are the spermatogonia. And then look at all these spermatids. All of these are spermatids. And then these are the sperm. See how they're getting longer and smaller? These are the these are the sperm. That's what I mean to say. These are the spermatids. Let me say it correctly. These are the spermatids, and these are the spermatozoa, the sperm, if you will. Remember, we're not going to be able to visualize the secondary spermatocytes. So it goes from spermatogonia, primary spermatocyte, to spermatid. It goes secondary, but we don't see it, and then to sperm as you go towards the interior. And then, of course, this is a very nice look at a spermatogonia, a Sertoli cell, get my words right, a Sertoli cell. See the nucleus, and you can sort of see a faint outline of the elongated cell there in this particular view. Here's another one. Sort of have to imagine the full length there, but you can see the nucleus and the nucleolus. You sort of have to imagine the sort of the columnar shape of the rest of the cell or the cytoplasm. And so we're seeing these fairly clearly there. Here's another look with the spermatogonia here toward the outer portion. All these are spermatogonia that I'm kind of indicating. Now we go a little deeper, and you see where we have these little stringy-like chromosomal structure. Those are the primary spermatocytes. And then deeper to that, we would get some spermatids. We're not really seeing spermatids or sperm in this particular view. Here we are, though. Look at this nice Sertoli cell there. Here's another one. Here's another one. And these are primary spermatocytes, aren't they? Well, what are these? Well, these are your spermatogonia. Here, here, here. And then these are your spermatids. And then these are your sperm cells. So we can see these quite nicely. This is another look at it with a more typical H&E non-plastinated view. And again, to your outer edge, those are spermatogonia, and then you can see the numerous primary spermatocytes there, here and here and here and here, and a bunch of them. And then your spermatids are here, 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 all these. And then see where they become little dots or kind of elongated dots. Those are your sperm. And they're in the process of forming a tail, a flagellum, but they're still in the process. This out here, these rounded cells with the nucleus and a rather eosinophilic cytoplasm, these are your interstitial cells of Leydig, which produce your androgens or your testosterone. You also see the myofibroblasts, these flattened cells along here. You see those as well. Here's a little bit of collagen and the like.
For us, are we gonna be using spermatozoa and sperm kind of like interchangeably?
Yeah, interchangeably.
Okay.
I know that's a little confusing. Sperm is just a short for spermatozoa.
Okay.
Yeah, so that's exactly right. Yeah. It's good to get those things cleared up, because otherwise you think, was there like another step in there or something? It can easily get confused by something like that. This is a scanning electron micrograph showing it. This is the maturation of the sperm, spermiogenesis. Remember, it has an acrosomal head, which is going to allow the digestive enzymes encased in it when it interacts with the egg and the outer parts of the surrounding the egg. There is an acrosomal reaction, and it allows penetration of the sperm into the egg. And then you have the mid-piece with regular organelles, and then the flagellum. Here's those structures again. This is showing the spermiogenesis, as it is. This is from your book as well, this diagram here. This shows it with an electron micrograph, that process. Here's the acrosomal membrane. Plasma membrane's kind of pulled off there. That's what that is. Okay. Within the flagellum, the tail of the sperm, we have microtubules that are made out of actin. And they have the 9+2 arrangement, a 9 doublet with microtubules in the center. I mean a 2, boy, I have a hard time. 2 doublet here, but a 9 out here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2. That's the 9+2 microtubular arrangement. This is a sperm with the flagellum. Now this is some more pictures of the seminiferous tubules, so we get a lot of practice here. And oh my goodness, we're about done, aren't we? So here's the Sertoli cells and all that. I think you, without much practice, you'll be able to get these pretty well. These are easier with it. This is a plastinated, it's put with a plat, so that's why it looks really pop. They're not gonna look as good in lab. But when you see the shape so clearly, it helps you when you see it more like this, which is more reality for us, okay? So we're gonna be looking at this. But you can already tell, see the stringy part here? Those are the primary spermatocytes. And, so on the edge you have the spermatogonia. So we'll see it. It's just, it looks more like this than those. This is nicer looking. But we're gonna have some digital questions from these nice looking ones too, so that'll be helpful, hopefully. Okay. Just going to show you, here's a list of all the functions of the Sertoli cells that you can look at, and the functions of testosterone. So, having said all that, tomorrow we'll do the rest of this, and we'll be doing this in lab on Wednesday. So what I really mean is Wednesday we'll be doing the rest of this. Tomorrow in lab, we're gonna be doing respiratory and urinary. For urinary, it's basically the kidney, the ureter, the urinary bladder. We might peep at the urethra, or we could leave that until the reproductive system, but we might peek at it. So there's probably about 4 slides to look at under the kidneys, or the urinary. And then for the respiratory, well, basically we look at the trachea and the vocal cords, and we look at the nasal area and the olfactory. Then of course we look at the lung. So that's kind of what we'll be looking at tomorrow. Okay, thanks so much.